Good morning, everyone. This is Dr. Sanjeev Pandey, and you are watching SP at Bio. Today, we are going to learn about regulation of gene expression in prokaryotes, particularly the Lac Operon. So, before going into the details, we should first know what are genes and operons. As told in many different video lectures earlier, the genes are the segments of DNA within the chromosome that produce a functional RNA. So this is the latest definition. Before this, scientists used to say that genes are the segment of DNA that produce functional proteins. But this definition was not a complete definition because those are also genes which synthesize RNAs like rRNA, tRNA, and other different kinds of RNAs. But these are not the protein coding RNAs. However, they are genes. So this definition is more correct, that the genes are the segments of DNA that produce functional RNA. So what are operons? In bacterial system, genes are present in groups. In fact, the resolution of genes in the chromosome is very high. They are very closely located, and many different genes are grouped or clustered, and they are regulated by a single regulatory element. Many times different groups of genes are regulated by a single regulatory element. Those are together called regulon. This operon is a single clusters of more than two genes, more than two genes, regulated by a single regulatory element. What do I mean by regulatory element? By regulatory element, we mean that a promoter, an operator, and a gene that may produce a repressor or inducer, repressor kind of protein. So this structure, some structural gene associated with some upstream regulatory elements like promoter, operators, and this repressor, this structure is called operon. This is an operational unit rather. When we add on as a suffix, it means an unit. So it is an operational unit. It acts like an operational unit because one regulatory element controls the transcription of two or more genes at a time, and a polycistronic mRNA is produced. Anyway, now let's understand that genes can be of two types. Those which are always expressed, they are called constitutive genes. They are constitutively expressed, they are housekeeping genes in other sense. And the others are regulatory genes. They depend on the environment, that whether they should be expressed or not, they depend on the condition. So constitutive genes are housekeeping genes. They has to be expressed all the time, otherwise the cell will die. For example, genes for essential metabolites, synthesis of essential metabolites, their anabolism, catabolism both may be included. And suppose the central metabolic pathway, the glycolytic pathway, the Krebs cycle, the lipid biosynthesis or breakdown, amino acid biosynthesis and breakdown, nucleotide biosynthesis, all these are essential components of a cell, and absence of a single metabolite will be dangerous for the cell survival. So genes associated with the synthesis or breakdown of these molecules are essential genes, and they has to be expressed all the time in all the cells. Those are called constitutive genes or constitutively expressed genes rather. Regulatory genes, as we were told, that they are conditional. They depend on whether the environment requires their expression or not. They could be of two types: inducible and repressible. Inducible means they were in negative control, negative mode, and they need to be induced. When the environmental factor arises, then these genes need to be switched on. So earlier they were in switch off mode. For example, in case of Lac Operon, which is a inducible operon, It is the inducible operons are also catabolic operons because they usually catabolize certain substrate, for example, the lactose. So when lactose is absent, the operon remains in switch off mode. When lactose is added, it is switched on. We will see it in our later slides. Repressible operons are, on the other hand, they were running. They were in switched on mode. They were in positive mode. So when the particular product becomes excess, then it goes into switch off mode. It is repressed. So it is basically anabolic operon. Example of anabolic operon is the tryptophan operon, trip operon. We will talk about that in next video. So let us see a little bit more about this difference between the inducible and reversible operon. So when the metabolite is present, the inducible operon will be switched on. When the metabolite is absent, it will remain in switch off mode. For example, Lac Operon, the metabolite is lactose. So as soon as the lactose is added, the genes for its breakdown, the genes for those enzymes which are required for its breakdown, like the beta galactosidase and also the Lac permease, which transports the lactose into the cell. Expression of those genes will start as soon as the lactose is added. And so here we can see that before that the level of those enzymes were zero, were static, and suddenly when you add lactose, the growth, the amount of those enzymes, the activity picks up. In case of repressible operon, when the metabolite is present is in excess amount to a certain level, then what happens? The operon goes in a switch off mode. The example is tryptophan operon, trip operon, substrate, the product, the metabolite is tryptophan. So here is graphically it has been shown that the activity of the enzymes responsible for the synthesis of tryptophan. As we talked that the repressible operons are anabolic operons, a synthetic process, a biosynthetic process. So when the amount of the product of this biosynthetic process increases a certain level, then the enzymes are not required anymore. The product is also not required in that amount. So the product goes and binds with an activator, and it inactivates that, and so that the production of the enzymes suddenly falls down. So this is called the operons responsible for the synthesis of that product are suddenly switched off when the product becomes excess, tryptophan level more than sufficient. When it becomes more than sufficient, it is repressed. The operon of the tryptophan is repressed then. Now let's move into the discovery of Lac Operon, how it was discovered. The Lac Operon was the first to discover, and the scientists which discovered them is the scientists that discovered Lac Operon are Francois Jacob and Jacques Monod. In 1960, they published a paper in Proceedings of French Academy of Sciences, where they talked about the cluster of genes that are regulated by some upstream elements. These genes are the beta galactosidase, beta is missing here, beta galactosidase, which clips lactose into galactose and glucose, and the Lac permease. They were situated one after another and were regulated by one regulatory element, promoter and operator. Later on, the third gene, the transacetylase, was also found to be associated with the same operon. So this concept it came into the field of molecular biology in 1960, after 1960. This is called operon model. The terms operon and operator were first introduced in this paper, and they did not use lactose as a substrate, rather, or inducer. Lactose is inducer as well as substrate. It is the natural. The Lac Operon is for breakdown of lactose, but instead of using that, they used another compound called IPTG, isopropyl beta D thiogalactoside. This is an inducer, but it is not a substrate. The substrate is this one, X-gal, which is 5-bromo-4-chloro-3 indolyl beta D galactopyranoside. So they use a combination of these two, which is the equivalent of lactose. This kind of molecules which are not a substrate but an inducer are called gratuitous inducers. So this is the story of discovery of Lac Operon. So how the Lac Operon is comprised of, what is the structure of Lac Operon? So this is a bacterial chromosome, part of a bacterial chromosome rather, where you can see that the lactose operon this is the site for lactose operon, which is situated between pure E and pro A genes, and there are other genes also on the chromosome. And if you magnify this site, Lac site, it is comprised of 3 structural genes, Z, Y and A. You may also call Lac Z, Lac Y and Lac A. Z is responsible for beta galactosidase synthesis, Y is responsible for beta galactoside permease or Lac permease synthesis, and the last one, A gene, is responsible for beta galactoside transacetylase synthesis. These are 3 structural genes consecutively present one after another and regulated by a single regulatory element comprising of operator and promoters. There are 3 operators over here, O1, O2, O3, but when Jacob and Monod described it, they described only one operon, that is O1. Later on, these O2 and O3 were also found to be the part of this system. And upstream to that is a gene, I gene. This is the promoter for the I gene. I gene synthesizes a protein, a tetrameric protein called, that is the Lac repressor. So this is part of the operon, Lac Operon. So the regulatory element comprises of the promoter, operator and the I gene, and the structural genes are Z, Y and A. This entire structure from PI to the stop codon of A gene is operon, is Lac Operon. So now let us see, this area is very, very important, and let us magnify that area, that how these things are present in the system. So basically the operon, let us first understand the promoter region. Promoter region is quite big, this one, and the 3 operators, the O1, O2 and O3. The O1 and O3, some part of them is overlapping with the promoter. O1 has this part overlapping, O3 has this part overlapping with the promoter. The promoter region binds with the RNA polymerase and also a positive regulator that is called CAP-CAMP complex. CAP is for catabolite activator protein and CAMP is cyclic adenosine monophosphate. When they bind here, then only RNA polymerase can bind at this position and transcription will start. The repressor will bind at O1. If repressor binds here, then RNA polymerase will not be able to bind because some part of it is occupied, and it will stop, it will block the binding of RNA polymerase. The repressor will not only bind with O1, it will also associate either O2 or O3. We will see it later. So this is the structure of the regulatory element of Lac Operon. So this is what is Lac Operon is, the structure of Lac Operon. Now let us see how lactose is metabolized, because we are talking about lactose metabolism. So as soon as this is a bacterial membrane and this is Lac permease, or you can also say galactoside permease, product of the Lac Operon second gene. It is a membrane bound transporter. So when lactose is added in the medium, it has to be transported into the cell through this Lac permease, and inside the cytoplasm it is first converted with the help of the same enzyme beta galactosidase, this triangular molecule, into another compound which is the actual inducer called allolactose. In allolactose, the galactose and glucose are bound by beta 1,3 glycosidic bond, whereas in case of lactose, they are attached with beta 1,4 glycosidic bond. Both of these compounds, they are disaccharides. They will be broken down into monosaccharides, galactose and glucose, by beta galactosidase again, and that will further enter into the central metabolic pathway to produce energy. So this is how lactose is metabolized. Here this molecule is important. We will see that the lactose, when converted into allolactose, this molecule binds with the Lac repressor and inactivates it. So now let us see how gene operon regulation, Lac operon regulation occurs in. So the first condition is when lactose is absent, when the gene is in negative mode, switch off mode. How it is in switch off mode? The LAC-I gene, it transcribes an mRNA that produces a tetrameric repressor, that is called Lac repressor, in absence of lactose. And it goes and binds with the operator region O1 and also associates the O2 and O3 regions. So the RNA polymerase cannot bind here and transcription does not occur. This is what is negative control. Now when lactose is added in the medium, then what happens? A basic amount of beta galactosidase is already present, and that converts lactose into allolactose. How a basic amount of beta galactosidase would be present? This is because when the cell is dividing at a very high rate, when the cell division rate is faster, because of the nutritional availability, then what happens? These molecules, Lac repressor, becomes diluted in the cell, and there are fractions of seconds when this is not bound here, and during that period the RNA polymerase binds and it transcribes a certain amount of these genes from where the beta galactosidase is. This is called sneak expression. Okay, sneak synthesis of beta galactosidase that goes on at a underground level. So that converts the lactose into allolactose. Allolactose binds with this Lac repressor and it inactivates it by changing its conformation. Now this Lac repressor is not able to bind on the operator region. Instead, the RNA polymerase binds over there, and the binding of RNA polymerase is dependent on the binding of cap-cam complex also. Until and unless cap-cam complex is bound here, RNA polymerase cannot bind properly, efficiently, and the transcription will not occur efficiently. It will only be 2% of the total transcription if cap-cam complex does not bind. So binding of cap-cam complex is very, very essential for Lac operon to be completely expressed. A polycistronic mRNA is thus produced. From there, 3 different products, beta galactosidase, Lac permease, and transacetylase are produced. So this is called positive control. So now let us move into think why cap-cam complex is so necessary. What is catabolite repression? What happens if cap-cam complex, when cap-cam complex is not formed? Under what condition it is not formed? If glucose will be present in the medium, then the level of CAMP is so down that it cannot associate with cap-cam complex. There is no CAMP rather in the cell. It is decreased to a level, and hence the cap-cam complex is not formed, and Lac operon is not, it remains in almost in a switch off mode even when the lactose is present and the Lac repressor is inactivated. This situation is called catabolite repression or glucose effect, and it will give if you measure the growth curve of a culture in which both glucose and lactose are present, then what will happen? The growth curve will be divided into two growth curves merged together as if. This is called dioxy growth curve. There will be 2 lag phase, log phase, and a stationary phase and death phase. The first phase is for glucose. When glucose will be completely exhausted, then only expression of Lac Operon genes will start. The beta galactosidase synthesis will start and lactose breakdown will occur, and then the cell will again enter into a log phase after going into a short lag phase when the glucose is exhausted, because it will take time to express those genes and beta galactosidase to achieve a certain concentration. So a little bit of lag phase will occur after glucose exhaustion, and then again a log phase will occur when lactose is being metabolized. When lactose will be consumed completely, then again stationary phase and death phase will occur. This kind of growth phase is called dioxy growth curve, and it occurs because of the presence of glucose along with lactose in the medium. So how glucose actually decreases the concentration of CAMP? When glucose enters into the cell, what happens? It is immediately phosphorylated, and that is very necessary because once it enters into the cell, it should not be expelled out. So it is bound with a phosphate, which is a charged molecule. So any charged molecule cannot cross a membrane. So this glucose is immediately encapsulated, you can say. So how this transport occurs and what happens? How it is related with the level of CAMP? The transport of glucose, mannose, different kind of raffinose, all these kinds of monosaccharides is dependent in bacterial system on a system called phosphotransferase system, which is a cascade of reaction to transfer phosphate to the molecules. The process starts from phosphoenol pyruvate, which is converted into pyruvic acid, and it transfers the phosphorus to enzyme 1, from where it is transferred to a heat shock protein called HPR, and from there it is transferred to another protein E3, the designation of which has been changed. It is now E2C. Probably you should be checking it out. And this is also E2A, B, different names have been given. E3 has been changed. So let us keep it E3. It is an older designation, no problem. The concept is this: when E3 is phosphorylated, it has a capacity to transfer the phosphate to glucose, and it does so, and it immediately becomes dephosphorylated. A dephosphorylated E3 does following things, which is related with Lac Operon. The first of all, it blocks the transport of Lac Operon. It blocks the Lac permease, so that it blocks the Lac permease so that the transport of lactose is blocked. And again, it induces the activity of phosphatase enzyme, which converts CAMP into AMP. So level of CAMP becomes less here by this mechanism. Certain amount of CAMP is expelled out via CAMP pores, and this enzyme, which is responsible for the conversion of ATP to CAMP, the adenyl cyclase, that is inactivated by E3, dephosphorylation of E3. So these are the mechanisms. These three mechanisms decrease the concentration of CAMP, and this mechanism does not allow lactose to enter into the cell. So there is no question of lactose metabolism. So that this is how CAMP level is down and CAP-CAM complex is not formed and catabolite repression occurs. Now let us see several conditions of catabolite repression. You can say when glucose is present, glucose is absent, several conditions. Let us understand that this comes in question many a times. So this is the normal condition. When glucose is absent but lactose is present, then gene expression will be strong enough, will be complete. CAMP level will be high, CAP-CAM complex will bind on this cap site, and the RNA polymerase will also associate with that, with this carboxy terminal domain, and transcription will occur at 100% rate. If glucose is present, lactose is not present, then automatically Lac repressor will go and bind on the operator region. Glucose is present, so CAMP level will be down, so there is no question of binding of cap-chem complex here. When both of them are absent, then CAMP level is high, but there is no chance because lactose is absent, so Lac repressor will go and bind on the operator region. RNA polymerase will not bind here. When glucose and lactose both are present, then the situation we talked earlier, that CAMP level will be down, so cap binding site, the cap protein will not be able to bind here. Cap-cap complex will not be able to bind here. Even when lactose is present, the Lac repressor is inactivated. There is a proper chance of binding of RNA polymerase over here. There is no blockage, but because cap-cap complex is not being formed because of the decrease in concentration of CAMP, the transcription will not occur. It will occur at a very basic level, the level at which RNA polymerase can synthesize without cap-chem complex is only 2% of the total at that level. So now let's move into a different matter, that how when this Lac Operon was being deciphered by Jacob and Monod and later on by others, different mutants of E. coli were studied, Lac Operon mutants, because we know that the mutants always help us to understand the function of genes or function of segments of genes that are responsible for the expression of the genes. So for this you require a mutant bacterial cell, and you can compensate the or you can rescue the mutant cell with the help of a plasmid by supplying the correct form of that gene, the wild type form of that gene. So this kind of system is required, where the Lac I, suppose in this case Lac I is negative, this is the chromosomal DNA, so this is unable to produce Lac repressor, and this will be constitutively expressed if lactose is present or absent, but this will be expressed because Lac repressor is not being synthesized. But is it possible to compensate Lac I by supplying a Lac I plus in a different plasmid? Yes, it is possible, because Lac I synthesizes a diffusible protein which can be given by any gene in any DNA molecule. So it is a trans-acting factor. It does not need to be present on the chromosome, because it produces a diffusible substance that can bind at this position where operator is present, and it can block the expression of LAC-Z gene. But similarly, if other genes, Lac I is correct, but if P is mutated, if O is mutated, then we cannot compensate that, or we cannot rescue that with the help of a plasmid containing the wild type of that gene, because promoters and operators, they don't produce any diffusible substance. They are a structural part of the operon, they are DNA, and they has to be present in right position in the operon just before the structural genes. So supplying them in a different plasmid for rescuing will not be functional, because they are required at cis position. So they are called cis-acting factors. So this is what trans-acting and cis-acting factors is about. Now let us see different conditions of merozygotes. This is called merozygote because in bacterial system we don't get zygotes, diploids, complete diploids. Only a part of the genome can be supplied or present. So diploid is only for that region of the genome. So it is called partial diploid or merozygote. So here is a merozygote, I minus, I plus. Others genes are positive. So this condition. So in this case, what will happen if lactose is present? Then gene expression will occur. If lactose is absent, this gene, which is the correct form of the gene, will produce the Lac repressor, and this will bind on the operator and will block the synthesis. So no expression will occur, even when the actual I gene is mutated. If this had not been there, then the expression would have been constitutive. In the next mutation, this is called OC mutation. OC is a constitutive mutation. It is a mutation on the operator region which causes a constitutive expression of Lac Operon. If it occurs, the Lac Operon will always be expressed even when the lactose is present or absent, or OC is a dominant kind of mutation. It will in fact overcome all different kinds of mutations if OC is present. The gene, the operon, Lac Operon will be constitutively expressed always. Now let us see, when P is mutated and you have given a P, rescued P with a plasmid, in this case it will not work because P is required at cis position like O. So in absence of inducer or in presence of inducer, when my P is mutated, RNA polymerase will not be able to bind even when lactose is present or it is absent. In case of absent, already Lac repressor has bound there, so there is no question of expression. And there is a kind of mutation I minus D. This produces a defective kind of Lac repressor. So the Lac repressor that will be produced will be defective, and if you if you rescue it with an I plus wild type gene, sorry, then also this wild type gene will produce a correct form of the protein Lac repressor, but that will mix up with this defective protein and will produce a defective Lac repressor. So this Lac repressor will not be able to bind on the operator, and the expression will always occur, either the inducer is present or absent, whatever. But another mutation called IS, it is a super repressor mutation. This is a mutation in the Lac repressor on the site where the allolactose binds. So now allolactose cannot bind to Lac repressor. See, allolactose, if it does not bind, what will happen? The Lac repressor will not be inactivated. It will always remain active. So it will always block the operator region even when the allolactose or lactose is present. So it does not depend on the presence or absence of the substrate and will block the transcription. IS is dominant over ID and OC is dominant over all of them. So this is how these mutations helped us to understand the function of different regulatory elements. Now let's move into some details. When Lac Operon is being expressed, the binding of cap-cam complex is very important, no doubt. So how does this cap-cam complex associate or interact with the DNA? This interaction has been studied by X-ray crystallographic studies, and it has been found that the DNA bends like this when the cap protein binds with this DNA. It produces a 90-degree angle kind of thing. And this bending is very, very essential. Because of that, the RNA polymerase is able to bind with the cap, able to interact with cap protein with its carboxy terminal domain. The DNA bends like this, bringing the RNA polymerase a little bit closer to cap-cam complex, and the RNA polymerase can associate with its CTD, carboxy terminal domain, with the cap protein, and transcription will start then. That is why this cap-cam complex binding is essential. Otherwise, RNA polymerase will not be able to synthesize at its full length or full capacity. So this is the regulatory element, that part of the gene. I have attached this in this slide also to understand where these molecules bind. Now let's move into the next interaction, in which the Lac repressor, how the Lac repressor interacts with the operator region. There, when Jacob and Monod they talked about only one operator, that is O1. But later on O2 and O3 were also found. And this experiment shows that this is a synthetic DNA, 21 nucleotide long, and this is the Lac repressor, the 4 tetrameric structure. We can see that the Lac repressor binds with 2 such oligonucleotides, 21 nucleotide base pairs long oligonucleotide, suggesting that the Lac repressor binds with 2 regions, not 1 region, comprising of about 21 base pair. So how this binding actually occurs in the DNA? It occurs if O1 and O3 get associated like this, then what happens, the in-between part is lobed like this. So this is the Lac repressor, and this is the CAP-CAM complex, which becomes entangled inside this loop. So it cannot interact with RNA polymerase, and there is the transcription will not occur, of course. If the interaction is with O1 and O2 rather, then this loop will not be formed, rather a hairpin structure will be formed. That will also block the transcription. So this is how these operator and Lac repressor, they interact together, and very interesting. And it has been seen that the presence of O1, O2 and O3, all these three are required for maximum repression to occur. But O1 and any of the O2 or O3 can produce strong enough repression. O2 and O3 either of them should be present. Deletion mutant study have shown that. This is again the same structure, just to have a parallel idea about where the things, where this Lac repressor is binding. So this is my last slide, and now let us see what transacetylase, this third enzyme, is doing, because there is always the discovery of Lac Operon has been quite half a century has passed, but there is no clear-cut idea how transacetylase is functional in this Lac Operon. Probably it is functional in some other process. But during the transcription, what happens? This is the Lac Operon, and we can see that there are intergenic areas between the Lac Z, Lac Y gene, this one, Lac ZY, and between these two genes, Lac Y and Lac A. Stoichiometry has shown that the concentration of Lac Z is 3 to 5 times higher than Lac A. So why this is so? This is because when the transcript is produced from this, the polycistronic RNA, that is clipped at this Lac YA juncture. This is 65 nucleotide. This is 65 nucleotide long segment, and this is the structure of that area. You can see this is the Lac Y, the stop codon of Lac Y, the upstream gene, and this is the start codon of the Lac A gene. So this is 65 nucleotide hairpin loop like structure present between the Lac Y and Lac A genes, and this RNA, the polycistronic RNA, when it is produced, it is later clipped at this position by a RNase, that is called RNase P. RNase P clips here, and it separates this Lac A RNA. The half-life of Lac A RNA is very, very less compared, about 10 times less than the Lac Z and Lac Y RNA, and that's why the concentration of transacetylase is very, very low. So these are the areas where the RNA is also clipped by some other RNAs, like RNase TI. It clips on these areas where the arrows has been shown, and where the triangles have been shown, the RNase VI clips on those areas. So this is how the transacetylase is, the RNA of the transacetylase decreases in concentration, and also the amount of transacetylase is comparatively less than beta-galactosidase in Lac permease, and probably the transacetylase is used in some other process, not in Lac metabolism, lactose metabolism. That is very clear to us. So that was what about Lac Operon, and I think I have tried my best to make the things clear, whatever is known about Lac Operon till date. And if anything has left, you can discuss it in the comment box. You can give your feedback also over there, and we can have a fair discussion over there. If something has been missed, something has been, you think that it has been wrongly said, then you can, of course, you can criticize over there. And don't forget to subscribe the channel, share it, and like it, so that it becomes available to more people. And also click the bell icon, top right at the corner, you can see. Thank you very much.